Good afternoon. First of all, I would like to say thanks for the invitation, especially for me as a surgeon. I think there are not so many surgeons at the EULAR meeting recommending you a new treatment option for systemic lupus erythematosus. And it's my first EULAR meeting, I have to state. Okay, the title of my speech is Alteration of Nucleosome Structure and Amelioration of Murine lupus by topoisomerase I inhibitors. I have no disclosures. First of all, in the beginning, I would like to explain how I came from cancer research to autoimmunity, or alternatively, how I came as a surgeon into SLE. Many years I worked in cancer research, and about 7-8 years ago we had an unusual animal experiment. We used immunocompetent mice. They were treated with a protein, with a human protein, and one group was treated only with a human protein and the other in combination with a topoisomerase I inhibitor, Irinotecan. What was happening during the second cycle of treatment, all mice which were treated only with a human protein died within 2 or 3 days. And in contrast, all mice treated with a human protein in combination with Irinotecan survived. And many of you know animal experiments, and they know that you normally don't have black and white effects. It was so interesting, this fact, that we thought we have to continue here. And what was happened? So we hypothesized that we sensitized these immunocompetent mice by the application of this human protein, so that we induced probably cross-species specific antibodies. We couldn't prove it because all these mice were dead. The second hypothesis was that the addition of Irinotecan prevented this hypothesized fatal hypersensitivity reaction. And the last hypothesis was, third hypothesis, that probably the topoisomerase I inhibitor, Irinotecan, might have useful effects in fatal hypersensitivity reactions like anaphylaxis or autoimmune diseases. So we went on with the story, and first we looked at anaphylaxis. Animal model inducing with off-albumin, there was no effect of Irinotecan at all. Then we implemented 2 models of spontaneous autoimmune diseases. The first was autoimmune diabetes in NOD mice. There was no effect at all with Irinotecan. And in the second model, we saw the dramatic effects in preventing the onset of spontaneous lupus disease. So, just I have to give a short introduction. What is topoisomerase I and what are topoisomerase I inhibitors? Because I believe that rheumatologists and nephrologists are not really aware of this drug. It's more a drug of used by the oncologist. So topoisomerase I is an ubiquitously expressed protein. It's really highly conserved in the phylogeneticity, and it is responsible for releasing torsional stress from the DNA. To achieve this, topoisomerase I binds to double-stranded DNA, induces single-stranded DNA breaks. Then the overbinded DNA can unbind to release the torsional stress. This is followed by religation of double-stranded DNA by topoisomerase I and by the release of topoisomerase I from the DNA. You can see that. So you have relaxed DNA. This is running in an agarose tray much more slowlier than supercoiled DNA. So how about the inhibitors of topoisomerase I? As I showed now, before topoisomerase I binds to DNA, induces single-stranded DNA breaks, and to this complex, known as cleavable complex, Irinotecan can bind because topoisomerase I has a conformational change after binding to the DNA. and irinotecan, when it binds to this complex, it stabilizes this complex so that the religation of the DNA cannot occur anymore. So what is the consequence for the cells? In rapidly dividing cells, such as cancer cells, this complex of DNA, topo1 and irinotecan can collide with so-called replication forks. And this collision induces double-stranded DNA breaks, and double-stranded DNA breaks are a hallmark of apoptosis cell death. That's why irinotecan is used as chemotherapeutic agent. In non-dividing tissue, there's a complete different situation. So we don't have these replication forks, and therefore we induce just single-stranded DNA breaks which cannot be religated. This inhibits proliferation but does not induce cell death. And there's another possibility, but it's connected to the second one, that we inhibit, that the topoisomerase 1 inhibitors, they inhibit DNA relaxation. The topoisomerase 1 inhibitor irinotecan, it's a known drug since many years, 15 years. It's approved by the FDA for the treatment of metastatic colorectal cancer. It has, in the doses used for chemotherapy, has side effects such as diarrhea and a neutropenia of short duration. So it's an old drug which is used for many years in the clinics. So this was long introduction. Now I come to our data. We started with this, I mentioned already, with a spontaneous model of lupus in New Zealand black, New Zealand white mice. And we started with a prevention and with a high dose. So that means we gave doses of irinotecan which were similar to the chemotherapy in the early age of these mice. So we started at week 13 before the onset of the disease. Normally in these mice, they develop signs of lupus disease after week 20. But what we can see here, when we gave irinotecan, so the saline mice, they develop lupus nephritis shown as proteinuria, but both irinotecan concentrations, again I say it's high dose, they completely inhibited the induction of lupus nephritis. You can see here the survival. We had a median survival of both irinotecan groups of 90 weeks compared to 36 weeks of saline-treated animals. We also started, because it worked really nicely, we started to treat the animals much more later, so starting from established disease. This is around week 35, so we measured proteinuria, and when they showed proteinuria grade C or grade 4, it's a high-grade proteinuria, we started to treat them with irinotecan. And as you can see here, in the saline-treated group, there's no reversal of proteinuria, but both irinotecan treated groups showed a reversal of existing proteinuria. We published this data in Journal of Immunology in 2010, but we were highly criticized by rheumatologists, nephrologists, and their arguments were, okay, we don't need another chemotherapy for young women, so this is not necessary. And I like this data, and to prevent my depression of my own, so we started to lower the dose, to decrease the doses of irinotecan, and we treated, here's the survival of these mice treated from established disease. Okay, and here you can see the data where we treated established disease with extremely low doses of irinotecan. The doses we used here were more than 50 times lower than the doses used for chemotherapy. And you can see here, as shown before, for the high doses, that even that low dose, extremely low dose of irinotecan reversed existing proteinuria. And more important, the survival of these mice was highly significant better of irinotecan treated mice compared to saline treated mice. Again, to say we started with a high dose and we started with a prevention study, we came up with a very low dose, 50 times lower in established disease. From the clinical point or from preclinical point, I think exciting data. But how about the mechanism? I have to show this mechanistic slide again. In the very beginning, we saw that probably we have a pro-apoptotic agent, and probably we induce apoptosis in B and T cells, and therefore we have an immunosuppression, and this might be the mechanism via the induction of apoptosis, and we looked at it. We looked at apoptosis in the kidneys of these animals, and here's a tunnel staining for anti-double-stranded DNA breaks. You can see here in saline-treated mice, you have a lot of cells with apoptosis or a high extent of apoptosis, but surprisingly, the kidneys of irinotecan-treated mice, they had much lower extent amount of apoptosis. We also measured caspase-3 activity, which is more specific for apoptosis, and we could see the same. So it was not apoptosis. In line with that, we looked at the number and activity of B cells, and we could see that there was some immunosuppression, transient immunosuppression when using high-dose irinotecan, but here is with a low-dose irinotecan is showing here and here. There was no immunosuppression anymore, but these dosages of irinotecan were highly sufficient to suppress lupus nephritis, though there was no relevant immunosuppression. And in line with that, the amount of anti-double-stranded DNA antibodies in the sera were not decreased, even in the high-dose irinotecan group. There was no difference in the levels of anti-double-stranded DNA antibodies. So what alternative mechanisms should have been? And we looked furthermore, and we speculated for a very long time that probably the induction of single-stranded DNA breaks might be responsible. So the explanation was, or the hypothesis was, okay, we have anti-double-stranded DNA antibodies in lupus, but we induce probably single-stranded DNA breaks, and then these antibodies, they can't bind anymore, and it would be an easy explanation. Many years ago, Martin told me, or some years ago, told me I'm wrong, and you are right, I was really wrong. It's probably not the single-stranded DNA breaks. There might be another possibilities that it could be related to the extent of relaxation of the DNA. Because if we might inhibit DNA relaxation, might that alter the binding of anti-double-stranded DNA antibodies, and it was indeed like this. These are in vitro experiments. We use different forms of DNA. This DNA was in vitro incubated with recombinant active topoisomerase 1 inducing DNA relaxation. And then we measured the extent of anti-double-stranded DNA binding. And as you can see here, when we treat the DNA with increasing amounts of topoisomerase 1, then we have more relaxed DNA, we have more anti-double-stranded DNA binding to this DNA. This worked with antibodies from lupus-prone mouse and from patients with SLE. We looked at other DNA, so it's in the title of my speech, the nucleosomes. We looked for nucleosomal DSA as well, and there we had the same effect. When we treat the DNA with topoisomerase 1, we relax the DNA, and we find a more binding of anti-double-stranded DNA antibodies. And we used a supercoiled DNA, which can is used for these experiments. You can see it nicely on an agarose gel, which is not the case for nucleosomal DNA or calf-thymus DNA. You can see here the supercoiled DNA running faster, and here the relaxed DNA, and here when we treat this the supercoiled DNA with increasing amounts of topoisomerase 1, we have more relaxed DNA, the more we have anti-double-stranded DNA binding. This effect we were able to inhibit with a topoisomerase 1 inhibitor. I cannot show these data due to the time restriction. I want to say thanks to the people of my lab, Selina, technician, Manuela, the postdoc, and Andreas Keil, my PhD student. He will finish his PhD this summer. Excellent thesis. I want to just show another 2 slides because there were a lot of in vitro data and animal experiments, but I think this has no sense. Just it's academic knowledge just for nothing, and therefore I started to create protocols for a first clinical trial. I called it LUNERI-1 because the protocol is very similar to the LUNAR study. And so we want to treat patients with active lupus nephritis refractory to conventional treatment with low-dose Irinotecan. We will see whether this has an effect. And there's the protocol. I don't want to go in detail, just to show that we want to end up, if it goes good, we want to end up with Irinotecan as a single-agent treatment for patients with refractory lupus nephritis. And to obtain the funding for such a study, I founded a non-profit organization to get donations to hopefully start a study soon. Thank you very much for your attention.
Thank you for this beautiful presentation that is now open for discussion. No question?
Could you just remind me, what are the side effects of Irinotecan if you treat cancer patients?
Excuse me. Side effects are, so it's diarrhea, but it's normally manageable now, and neutropenia of short duration. So there is some immunosuppression, but I think it's really in the high dose used for Irinotecan. So we would plan to use much lower doses for patients with lupus nephritis.
Yep.
Frédéric Houssiot from Brussels. You did not show the pathology effects of your drug on the mice kidneys.
Excuse me?
You did not show the pathological changes induced by your drug. That is my first question. And the other one, in the LUNERI protocol, you are going to use a no-steroid regime?
No. In the beginning...
That would be interesting.
No. In the beginning, we would start a treatment with a conventional medication. Okay. So in the first 4 months, then we would have another 4 months where it could be stabilized. So and after 8 months, we would plan to reduce conventional medication. Right. No, not in the beginning.
And what about the pathology in mice?
Excuse me? Yeah, pathology in mice. So we showed it already in the Journal of Immunology paper. So I just didn't show it because we have an amelioration of the kidney score very nicely shown. Yeah.
And no chronicity.
Excuse me?
And no chronicity.
No. Okay. I mean, what we see when we don't give anymore the Irinotecan, the disease comes back. So it's but. We can suppress it, but we cannot forever.
Maybe I missed it, but if you stop treatment of the NZB and the W mice SLE model, will this be followed by a flare?
So after when we treated established disease, so we waited until the proteinuria came back. So, and then we started treatment again.
And when you stopped And then the proteinuria improved again.
Yeah, so after, it depends, after 6, sometimes after 12 weeks came back, yeah.
Thank you for the talk. Do you know if Irinotecan has to go to the nucleus or it can work outside of cells?
That's a very good question. We are discussing it many times. We looked for topoisomerase in the sera, but we were not. We spent quite half a year for that. We were not successful. So probably the mechanism is like that something will takes place inside the cell in the nucleus, and then this modified DNA is getting released. This is more likely than it will really happen in the sera, that in DNA relaxation or something like this might be not happen in the sera.
One more.
Yeah, of course.
Does it work without Topo 1? Does it work without the enzyme, topoisomerase 1? Does Irinotecan do the inhibits the relaxation without topoisomerase 1?
No.
Thank you very much indeed.